In 1987, a team of Japanese scientists was studying a common bacterium, and they stumbled upon something odd, a stretch of DNA with strange, repeating sequences. They had no idea that these seemingly random repeats would one day become the most powerful tool in modern genetics. For years, these repeating sequences, known as CRISPR, sat as a curiosity in microbiology. Scientists weren't sure what they did or why they existed, but the mystery would eventually unlock a revolutionary technology. It took about 2 decades before researchers began to piece it together. In the early 2000s, scientists realized that these CRISPR sequences matched the DNA of viruses that attack bacteria. It was a clue. The breakthrough came when they understood that CRISPR is part of a bacterial immune system. When a virus infects a bacterium, the bacterium captures a piece of the virus's DNA and stores it in its own genome, like a wanted poster. If the same virus attacks again, the bacterium uses that stored DNA to recognize the intruder and chop it up. It's a kind of genetic memory passed down to future generations. The key was a protein called Cas9. Cas9 acts like a pair of molecular scissors guided by a piece of RNA that matches the viral DNA. It can cut, edit, or delete specific genes. In 2012, 2 scientists, Jennifer Doudna and Emmanuel Charpentier, showed that this system could be reprogrammed to cut any DNA sequence they wanted. They had created the world's first programmable genetic scissors. The discovery was a turning point. For the first time, scientists could edit genomes with incredible precision, speed, and affordability. The door to a new era of biology had swung wide open. CRISPR was quickly adopted in labs around the world. Researchers began using it to modify crops for better yield, create models of human diseases, and explore new therapies. One of the most exciting potential uses is in medicine. Scientists are investigating whether CRISPR can fix genetic mutations that cause diseases like sickle cell anemia and certain types of blindness. But the technology also raises serious ethical questions. The ability to edit human DNA, especially in embryos, challenges our understanding of what it means to be human. In 2018, concerns became reality when a Chinese scientist, He Jiankui, announced that he had used CRISPR to edit the genes of human embryos. The twins, Lulu and Nana, were born with altered DNA. The announcement sparked global outrage and scientific condemnation. He Jiankui had crossed a line that many felt had not been ethically prepared for. He was later sentenced to 3 years in prison, and his research was widely rejected by the scientific community. The incident showed that the power of CRISPR comes with great responsibility. Despite the controversy, research continues at a rapid pace. New versions of CRISPR are being developed that are even more precise and can edit DNA without cutting it. Scientists are also finding ways to use CRISPR to target diseases in adults, potentially curing conditions that were once considered untreatable. In agriculture, CRISPR is being used to create crops that are more nutritious, resistant to pests, and able to thrive in changing climates. This could help feed a growing global population. But the technology is still young, and there is much we don't know. Off-target effects, where CRISPR edits the wrong part of the genome, are a serious concern. Scientists are working hard to make CRISPR as safe and reliable as possible, but the potential is enormous. If used wisely, it could alleviate suffering and transform our world. The story of CRISPR is a reminder that great discoveries often come from unexpected places. A strange pattern in bacteria has become one of the most promising and controversial tools of our time. As we continue to unlock its possibilities, we must also engage in the difficult conversations about how to use this power. The choices we make will affect generations to come. What began as a curious observation in a Japanese lab in 1987 has evolved into a technology that could redefine life itself. The future of CRISPR is being written now, and it's up to us to decide its story. Subscribe for more stories like this.